Hello, welcome to this session on ataxia mouse models. Well, we're glad that you could spend the next hour with us learning about mouse models and why we use them for ataxia research. And with that, it is my absolute privilege to introduce our speaker today, Dr. Harry Orr. He's a geneticist specializing in neurodegenerative diseases. He was born in Virginia and grew up in Michigan, where he received a Bachelor of Science in 1971 from Oakland University. He did his Ph.D. in neurobiology at Washington University in St. Louis, Missouri, where he studied the retina. In 1980, he completed his postdoctoral fellowship at Harvard University in the laboratory of Jack Strominger, where he studied the genes encoding major proteins of the immune system. In 1981, Dr. Orr joined the University of Minnesota Medical School in Minneapolis, where he is now a professor of genetics in the Department of Laboratory Medicine and Pathology and the director of the Institute of Translational Neuroscience. His work on genetics and the immune system led him to a collaboration with Huda Zoghbi to the discovery of spinocerebellar ataxia type 1, which we're very well familiar with on this call, but for folks who have not heard of it, it is a neurodegenerative disease affecting motor coordination and cognition. Uh, or continues to study SCOT1 and is working to translate those therapeutic discoveries to new therapies. And with that, Dr. Orr, the floor is yours.
Oh, thank you very much, Celeste. And thanks to NAF for the opportunity to talk to you about the use of mouse models in ataxia research. And again, I just would like to emphasize the point, if you have questions, please put them in the chat or the Q&A, and Celeste is in charge of getting them in front of me. And if you do have questions as we move through the talk about clarity or some issues, please don't hesitate. I've given her instruction that it's okay to interrupt me. So, speaking to you from relatively warm Minneapolis, a week ago we got 9 inches of snow, and I'm pleased to say that it's now all gone and working its way down the Mississippi River to the Gulf of, well, whatever the name of that body of water is now. Okay, here are my disclosures. You can see we have collaborations with several pharmaceutical companies. I have no financial interest in any of these, and our research is supported by grants from NIH and private donations. But perhaps my most important disclosure is that having grown up in Michigan and now at the University of Minnesota, I have frequent conflict of interest with Hawkeyes, Badgers, and Buckeyes. So here's sort of the 3, if you will, major points I hope to get through in the next few minutes. I want to talk about how we use mouse models of ataxia to elucidate disease mechanisms and pathways, and then transition from this sort of basic science, if you will, to hopefully identifying potential therapeutic targets. And then the long-term goal is to utilize these mouse models in what we call preclinical studies, the goal of which are to assess the efficacy and safety of these potential therapeutic agents. And this is an important step towards being designated an IND, or an investigational new drug, by the FDA. And it's an important step for now moving a potential therapeutic approach to clinical trials. Okay, a couple of points that we need to keep in mind are outlined on this slide. I hope everybody appreciates that on the left, the mouse brain is much smaller than the human brain, and there are specific regions of the mouse brain that, shall we say, are not as developed to the degree that they are in the human brain. We like to think of us as humans as being thoughtful and cognitive people, whereas mouse perhaps are just run around looking for food and whatever. The other important issue in generating a mouse model of a disease like ataxia, many of which are late onset diseases, is that the average lifespan of a mouse in our housing facility is about 2 years of age, whereas currently my understanding in the United States is the average lifespan of humans is around 77 years. So, back to the aspect of the relative complexities between the mouse and the human brain. The important thing about the organization of the mouse and human brain is that the region of the brain that's important for controlling motor function and balance, which is a major aspect of the symptoms that we see in the ataxias, i.e. the cerebellum and the other regions of the brain that the cerebellum is connected to, that are important for motion and control of balance, are highly conserved between mice and humans. So this is just sort of an outline, if you will, a roadmap of the cerebellar cortex. Many of you have probably heard about the central aspect of the cerebellum being these Purkinje cells or Purkinje neurons. They're very important in integrating the information about motion and balance. And then it's the output from the Purkinje cells down first to the cerebellar nuclei, and then subsequently other regions of the brain that are manifest in the role of the cerebellum in controlling aspects that are the dysfunction of which are important aspects of the various ataxias. Now, when we make mouse models of the ataxias, we are essentially making mouse models to two groups of ataxias, and these are what we call the inherited ataxias. On the left here are the recessive ataxias, which make up about 30% of the ataxias. And the important thing is that in a recessive disease, both parents, which are indicated here as being half colored in, are carriers of the mutation that causes the gene. And it's a fraction of their offspring, fully colored here, are the ones that inherit both of these each mutation, if you will, from the father and from the mother. And so the parents of an affected individual typically are not affected with the disease. In contrast, in the dominant diseases, the dominant ataxias, which make up only almost 60% of the ataxias, if you are affected with a dominant ataxia, it's highly likely that you have one parent that was also affected, and you only need to inherit one mutant gene from the affected parent in order for you then to be susceptible to this type of ataxias. Now, when I say your parent is highly likely to be affected, there's a group of the dominant ataxias, for example, SEA1, which we will talk about, but also SEA2 and 3, and a couple of others, where the mutation is this expansion of a DNA repeat, a CAG repeat sequence. And so there are occasions in some families where the parent has a length of repeats that's not quite long enough to generate the affected disease status, and so they're unaffected. But in the process of being transmitted from the parent to the offspring, the repeat goes long enough, a little longer, so that it's now in the affected range. Now, what's listed here is Friedreich's ataxia, which is probably one of the predominant recessive ataxias for which there are several mouse models, and we'll talk a little bit about the types of mouse models that have been generated for Friedreich's ataxia. And then on the right here is a long list of the dominant ataxias for which various mouse models have been made. And we're going to, in this case, we're going to focus, and the examples that I'm going to give you is some of the work that we have done in SCA1 and how we're using these mouse models to further our understanding of this form of ataxia and moving the research towards the treatment. And basically, the approach that is being utilized in SCA1 is being utilized for many of these other diseases. So let's talk a little bit about a mouse model, which I find particularly intriguing for Friedreich's ataxia. So Friedreich's ataxia is true of most recessive diseases where the underlying cause of the disease is the loss of function of a protein encoded by the affected gene. And so this disorder is caused by a deficiency in the protein encoded by the Friedreich ataxia gene, which we call frataxin. And so some of the mouse models that have been generated to replicate Friedreich's ataxia pathology and so forth are what we call deletion models, where the Friedreich's ataxia or frataxin gene has been deleted from the mouse. Friedreich's ataxia is also a repeat disorder. And so some of the other mouse models where people have inserted a long repeat into the mouse frataxin gene to, again, generate a mouse model of the human disease that we can then study. But in this case, Dan Geschwin and his group generated an what we call an inducible model of Friedreich's ataxia, where in the mouse genome or DNA, they placed a gene that if you give a certain chemical compound, doxycycline, to the mouse, this gene turns on, and the product of this gene then seeks out the Friedreich's ataxia RNA that comes from the gene and degrades that RNA. So it's an inducible, so we say, loss of function of the Friedreich's ataxia gene. And so what they did, and I'm just showing you two examples here, is what's shown here on the bottom of each slide is we want to focus on just two of the lines. One is the mutant mouse that was given this drug to target the frataxin gene for a short period of time. And then another group of mice, the drug was given continuously. So there would be a complete loss of frataxin in these animals over the period of the experiment. And this latter group of mice is shown here by this sort of purplish line. And you can see, for example, on the rotarod, which is a test we use to measure ataxia, these mice show a continuous loss of motor performance and balance and coordination. In contrast, the mice that were given the drug that induces degradation of the frataxin gene for just a short period of time are shown here in the tan line. And interestingly, after this drug is stopped and now the frataxin gene can come back on, you see that the mice are able to recover their motor performance. And over here, you see likewise, while the mice that have a continuous loss of frataxin show a continuous degradation in their ability to survive, the mice that get the drug for a short period of time and thus can recover show a restoration of survival. And so there's two important points. First of all, this links some specific characteristics of the mice to the expression of the frataxin gene. But from a therapeutic standpoint, this is really nice data to show us that mid-course in the disease, if we can correct and restore expression of the frataxin protein, we have a high chance of substantially improving the symptoms, if you will, of an affected individual. So this is one nice example of how mouse models of ataxia can be used, and they begin to help us understand when it's still efficacious to induce a therapy for a particular disease. Okay, so let's come back to this issue of modeling a human disease in a mouse, and specifically the average lifespan, if you will, that we talked about. And in this case, I'm taking our average lifespan of 2 years for mice and comparing it, say, most notably from many of the dominant ataxias. Typical onset is around 30 to 50 years. And so the challenge that we're faced with in the research lab is, how do we shorten the onset time in trying to generate a mouse model? We'd like to see the disease within 2 years, if at all possible, in order to have a valid model for a human disease. By valid, I mean one that we can go on and do additional experiments. So one approach that we can do is to overexpress the mutant human gene in the mouse. And in this case, we're talking about a mouse model that we made to understand how mutant ataxin-1 affects the function of these Purkinje cells, these critical cells in the cerebellum. And basically, you introduce this gene into the DNA of a mouse. You do it by injecting it into a fertilized mouse embryo. And the bottom line is the repeat is on the long end, but it's not out of the range that we see, for example, in the juvenile cases of SCA1. But because of how we've constructed this gene, it's really overexpressed compared to the expression level of the mouse ataxin-1 gene. So the first strategy that we can use to shorten the time span for developing disease into a 2-year time frame is to overexpress our disease-causing gene. The other strategy is that we can literally, by molecular approaches, introduce into the mouse ataxin-1 gene a length of DNA that encodes a really long stretch of glutamines. And these are CAG is a nucleotide sequence, and it's the length of the glutamines that determine the pathogenicity, if you will, of the protein. And so to get in this sort of model where now we're not overexpressing the mutant gene, because it's being expressed by the normal regulatory elements of the mouse gene, we're getting normal expression at the levels and spatial tissue distribution and temporal pattern, but we have a really long stretch of glutamines. So we either shorten the time frame by overexpressing a mutant gene or by putting in a really long stretch of glutamines, for example, in the case of the CAG diseases. And it's interestingly because work in Huntington's disease has shown that an interesting aspect of at least the Huntington's CAG repeat is that it goes through this process of what is called somatic expansion. So after being inherited to the offspring, it continuously grows over the years in many brain tissues. And once it reaches a length of 150 repeats, the pathogenic process, if you will, moves from something that takes decades to years to something that takes only months. And in fact, this is the time frame that we're trying to generate in our mouse models. So again, by putting in these long repeats in the mouse model, we're sort of bypassing this long period of time it takes for repeats in certain diseases to get long, and we're starting out right away with these long repeats so we can have a very rapidly progressing disease, a model that we can work with in the research laboratory. Okay, how do we look at the phenotype, which is the scientific word for symptoms of these animals? We talked about the rotarod, and this is an example, a cartoon example of the rotarod. It is literally a spinning rod on which you place the mice. You turn the machine on and it starts to go around and around, and the speed can increase with time. And you basically measure how long the mice can stay on this rotating rod. And coming from the northern part of the United States in Minnesota, sometimes I use the analogy, this is sort of like in a lumberjack festival, where you have lumberjacks are assessed by their ability to run on a log that's floating in a body of water. So they have to keep running faster and faster, and how long can they stay on that log rolling a task. The other measure we sometimes use is called a bar cross. It comes in various sizes, so you can increase the challenge of this task to the mice. And at one end of the bar cross, there's an open area, and mice very much don't like typically to be out in the open, so they'll go across this bar to get into this hidden chamber. It's where they feel more secure. And we measure time and how many times their feet may slip, or in dramatic cases, whether they fall off or not. Okay, so let me give you some examples of how researchers explore the pathogenic or the disease pathways using these various mouse models. And again, this is from our SCA work, and very early on, It was known in SEA1 that Purkinje cells are a major cell type that are affected in this disease. And so our
Sorry,
first,
Dr. Orr, could I
yeah,
interrupt, please? Just before moving on to this specific example, we have an attendee who's asking if you could explain broadly what a mouse model is exactly and why they're used for ataxia research.
Um, so a mouse model, if you in very general terms, is it's a mouse that is carrying a mutant gene that is known to cause ataxia, a certain type of ataxia in the human population. And so we call it we call it a model because it is a mouse that models the human disease. And I want to emphasize the word model. Um, it's because it's a mouse and because we have to do various things to generate the disease in a short time frame and so forth. It's what a real challenge facing researchers is understanding what characteristics of this mice replicate or mimic those that we see in humans versus what characteristics of these mice are more unique to the fact that we're modeling, if you will, the disease in the mouse. And I think the end point I would say is what these mice are is a very close, we think, biological system by which we can understand and hopefully identify treatments for the ataxias. Does that help?
I think so. Thank you so much.
All right. So back to our SEA1 mouse. And so two things we noticed, or that was clear that we noticed early on. One is that from human studies, these Purkinje cells in the cerebellum were a primary site of disease. And the second thing we noticed was that the ataxin-1 protein, the protein that's mutated in SEA1 in these Purkinje cells, resided very largely in the nucleus, the region of the Purkinje cells that contain the genetic information, the DNA for these cells. And so our first mouse model, one of the things that we did with this Purkinje cell specific model was to understand the importance of the mutant protein entry into the nucleus for the disease development. It's a very basic aspect of the molecular biology of this disease. And the way the ataxin-1 protein gets into the nucleus is a short stretch here at the end of the protein called the nuclear localization sequence. This is recognized by the receptors in the transport machinery that takes the protein from the cytoplasm of a cell and moves it into the nucleus. And so when we have the ataxin-1 protein with the intact nuclear localization sequence, this bright staining here shows you that the mutant protein has entered the nucleus of these cells. And down here we can see the mice expressing this protein in their nucleus develops a form of ataxia, i.e. imbalance and falling and so forth. If we now in the lab generate a mouse model where we have disrupted the function of this nuclear localization sequence in our mutant gene, place it into another group of mice, we see correspondingly the protein is not in the nucleus and the mice don't develop ataxia in Purkinje cell disease. So this tells us that in order for the disease to develop in these critical cells for the disease, the protein has to enter the nucleus. An important potential regulatory step in the development of the disease. But what we're showing here is in a mouse brain is a distribution of the expression of the normal ataxin-1 gene. And besides being expressed in the Purkinje cells of the cerebellum, it's expressed widely throughout the brain. And correspondingly, there's likely to be many other regions of the brain that are also affected in the disease. And how do we mimic this? Well, we go back to what we called our knock-in model, where we've inserted a stretch of long repeats into into an endogenous mouse ataxin-1 gene. And now we're looking at the mutant protein, if you will, being expressed normally throughout the brain and throughout the body. And now we can begin to understand what aspects of the disease outside of the cerebellum and the Purkinje cells are going on. And so let's come back to our NLS story and nuclear localization. So again, using our fancy molecular techniques, we can disrupt the function of this NLS now in our mutant gene that has these really long repeats. And what do we find? We find that on the rotor rod, here's the performance of the wild type mice. And you can see they stay on much longer than our mutant mice carrying the long repeat, which is shown here. And interestingly, if we prevent the entry now of this mutant protein from entering the nuclei of many cells, we see a substantial recovery on the rotor rod. We no longer see this disease, age-associated disease progression and deficit in their performance, but this manipulation alone is not sufficient to get full recovery. And specifically, there's an aspect of motor learning, if you will, that is not found even in these mice with the protein unable to get into the nucleus. So this begins to tell us that about the rotor rod phenotype, there's something more than the cerebellum and the Purkinje cells that are critical. And one of the things we're trying to understand using our mouse models is what other region of the brain might be contributing to this motor learning phenotype that we're finding altered in our mutant SCA1 mice. So back to the general idea of mouse models of ataxia, one of the important things is they can be used to understand exactly what regions of the brain are affected, and therefore where do we have to have our therapies distributed, if you will, such that we can get maximum efficacy in therapeutic potential. And just one other thing about these mice now that have this inability to get the mutant protein into the nuclei throughout many areas of the brain and tissues in the body in general, is that this has the most substantial impact on survival of our SCA1 mice. We get almost a doubling of their ability to survive by making this single amino acid change. So while entry of the protein into nuclei of cells doesn't cause a complete restoration, it really does cause a very substantial and significant improvement in overall health, if you will, of the animals, saying that this is a major aspect of this disease process. So the next thing I'd like to talk about is, and we sort of touched upon it, how do we begin to look at other regions? What regions of the body and the brain are critical for understanding these phenotypes or characteristics that we see in patients affected with ataxia? And this is understanding exactly what regions of the brain or what tissues in the body are important for the disease characteristics is really important in terms of not only understanding disease mechanisms, but also understanding where do we have to target our therapies to get maximum efficacy of improvement. So here's the strategy. It's another mouse model. And it's a knock-in mouse. So we're inserting human DNA into the mouse ataxin-1 gene. And what we've done is replace what we call the two coding exons, regions of DNA that encode protein in the mouse ataxin-1 gene with those from the human gene, and with a long repeat. So back to using this long repeat so that we can get disease phenotypes in the time frame of a mouse age. But the interesting thing in this process of replacing this mouse DNA with human DNA, we also flank our mutant region with what we call these LOX-N sites. And so why is this important? Well, first of all, what this does is it gives us a humanized version of the ataxin-1 gene in the mouse. So this is human DNA here. And so this is important for providing a preclinical screening platform, if you will, for therapies that are targeting the human ataxin-1 gene. The other thing is, from a research standpoint, we can cross these mice with a group of mice that express this enzyme called CRE. And what this CRE enzyme does is cleave at these LOX sites. And in the process of this cleavage, our mutant DNA from the human is deleted. And so depending on where this CRE enzyme is expressed in our mice, we can begin to characterize now what tissues are important for what aspects of the SCA or ataxia phenotypes. And so here's the example I want to give you. What we noticed is that in our mutant ataxin-1 mice with these really long repeats, as the mice age, here we're looking at an image from a normal wild-type mouse at 20 and 50 weeks. And again, our mutant mouse having the mutant human ataxin-1 DNA at 20 and 50 weeks. And look particularly as they get older, how they get this humpback, if you will. We call this kyphosis. And in mice, this is sometimes characterized by the loss of thoracic musculature, skeletal muscle. So using these mice, what we did first was delete now the mutant gene from muscle using this type of line. And you can see now this kyphosis has gone away. Whereas if we delete the mutant gene from neurons and neural cells in the brain, this kyphosis does not go away. And so what this tells us is that there's very likely a direct effect of the mutant ataxin-1 protein on skeletal muscle. So again, this might underlie the important aspect of SCA1, and that is the muscle wasting that's seen as the patients age. So again, it points to distribution of a therapy, not only in the brain, but if we want to also deal with this muscle wasting problem, we're going to have to think about a therapy that also is targeting the skeletal muscle. So the last thing I want to close with is how do we now use these mouse models to begin to assess potential therapies and their efficacy? And so, i.e., bring some of the work in research labs back to the patients. And what I want to give is a quick outline of experiments that we did a few years ago in collaboration with Holly Kordasiewicz and her colleagues at Ionis Pharmaceuticals. And what they had developed was what is called an antisense oligonucleotide drug. And this is a stretch of DNA that targets the mutant, that targets the ataxin-1 RNA, if you will, in affected cells and degrades it. So it's a way of removing, if you will, the mutant ataxin-1 from these affected cells and thereby hopefully restoring normal function. And because of the property of this drug, we have to inject it directly into the brains of the animals, specifically in the ventricles, so where the cerebral spinal fluid is, and then the ASOs distribute throughout the brain. So in this case, we're only targeting the brain itself and not the muscle. And what do we see? We see animals that if we inject with this drug, we see an improvement on the rotarod compared to the animals, the model that does not get injected with the ASO. And again, we see improvement on the beam cross. So in this case, here's how fast the wild type mice go. Mutant mice are much slower. Those mice that get the drug speed up and are very similar now. to what we see in normal wild type mice. So this sort of approach of targeting the ataxin-1 gene and reducing the expression of the ataxin-1 protein is now being evaluated therapeutically using several approaches, not only by Ionis, but several other pharmaceutical companies. Hopefully, in the not too near distant future, it will move into the clinical for a clinical trial, into the clinics. And finally, this is just showing you that this drug also improves the survival of our mutant mice. Here's the survival curve for the animals that received the drug, and here's those that did not. So that's what I want to talk about. I think what the major concept is that mice are a very important strategy for understanding how the various forms of ataxia develop and the molecular pathways involved with these disease, and then subsequently using this information for developing and assessing potential treatments.
Thank you so much, Dr. Orr, for a fantastic presentation. Already
Oh
coming
my
in.
goodness, there's 37 in the chat.
All good. So we're going to jump into questions right away. The first is, can mouse models be used to model any form of ataxia? We have a couple of different people who asked about specific forms like
Yes,
RSAX, AOA,
certainly, yeah,
GA3.
right. Any of the genetic forms. So either the recessive diseases like Friedreich's, there are mouse models, and then the dominant forms of the diseases like the SCA 1s, 2s, and 3s, and so forth. There are mouse models for all of these. Uh, I think that the other thing I didn't really talk about is that besides understanding therapies that are directed at these specific genetic forms of ataxia, these
Mhm.
are also models that can be used for understanding the efficacy of potential drugs that might be more symptomatic, that are treating the symptoms associated with ataxia, in which case they would potentially have broader implications and importance for ataxias in addition to the genetic forms.
That's very
So in
good
the
to know.
genetic models, we trying to develop therapies that are specific for those genetic forms, so more like cures, whereas we can also use them to look for drugs and therapeutic approaches that address the symptoms. So those would be more like treatments, if you will.
We did have a question about that, about if these mouse models are more so focused on treatments or cure development. So thank you for answering that in
Well,
advance.
the idea, the goal here is to get some good treatments, and then take that information and eventually move towards something that approaches being a cure.
And then we have a follow-up question about that. We have someone who's asked if there's a dominant ataxia medicine that is found, would that medicine work for recessive ataxias too? Perhaps you could talk about the difference between a genetic treatment or a symptomatic treatment and how they could be tested in mice.
Yes, it depends a lot on the various ataxias and the symptoms. Let's just say, for example, we had a drug that came out of the dominant research that was really good at restoring normal function in Purkinje cells across many different types of damage to those cells. That drug might be important for treating the recessive ataxias also, and maybe even some of the non-genetic forms of ataxia, given the importance of this neuron, these Purkinje cells, in the ataxia phenotype. But I think the reality is that at this point in time, our therapies for the genetic forms of ataxia are going to, at least the initial ones, are going to be very limited to that specific genetic form.
Thank you for explaining that. We've had a question around what kinds of treatments are tested out on mice. We've had people ask about stem cells, ASOs, supplements. So what kinds of treatments can you test in mice? Are there any limitations around
There
what
are
you can?
really no limitations, and so stem cells, cell transplantations, small molecules, genetic approaches where, for example, in the Friedreich's ataxia, one might envision putting in a normal frataxin gene that replaces, if you will, the mutant gene in the human. So it's a platform that's open to testing many different types of therapeutic approaches.
And then we have another question. Have in mouse models, which are different from human beings, but have researchers ever been able to reverse or cure ataxia in a mouse?
Um, I don't, I have a hard time using the word cure,
That's fair.
but certainly substantially mitigate the disease symptoms. Yes, we can see that we see that in many of the mouse models. Yes, with various treatments.
And then we've had a few questions around treatments that are now available in human beings and people wondering if these were ever tested in mice. So for drugs like troriluzole, were these ever tested in ataxia mice before they were
My
tested
understanding
in
is
humans?
yes, they were to some extent. Yes.
Could you speak a little bit about how mouse models could be used, for example, safety testing or efficacy testing before a drug is tested in humans?
Right. Certainly the FDA, my understanding is the FDA requires in the safety aspects of testing a potential treatment, the use of two species. And so typically the mice are one species that's used and you're just looking for adverse effects of the drug. But most of the safety testing is done in non-human primates. So which more closely mimics, if you will, or approaches what the situation might be in humans. And then, of course, the clinical trials are designed in phases, phase 1, phase 2, and phase 3. And the major goal of the phase 1 trial is, again, safety and tolerability. And then as that moves forward, and then you move into phase 2 and phase 3 to where you're testing the efficacy and the ability of the treatment to impact the symptoms.
We had a related question come up in the chat. If someone was asking if Skyclarys, also known as ovomalloxolone, was ever tested out in mice, it was. There's information available on the NAF website and the FARA website on some of the early work that was done using mouse
Mhm.
models to move things forward. We have a couple of questions from folks that are ataxia specific, but they're wondering if there's a mouse model for their type of ataxia. How would you recommend they could find this information? Is there a way they can look this up?
Um, yeah. There was a paper published in the Cerebellum a little while ago, focusing on the SCA1, so the dominant ataxias, and that has a table in it that lists the various mouse models for the different SCA1s. And I think perhaps somebody at NAF could get their hands on that table, and that would be a source, if you will, for which of the SCAs. I'm not that familiar with the recessive disease, other than to say there are several mouse models for Friedreich's ataxia.
All right, so you're assigning me a little bit of work. But if
Yes.
anyone on this call would like to contact NAF, just as such an individualized question, please send an email to research@ataxia.org. We'll help you find the information that you need. And then we have a different animal model question sneaking into the chat here. There's someone who is interested in your opinion on what might be a better model for ataxia research, mice or flies?
I think it depends on where you are. If you're at the beginning of the basic research and you're just trying to get at what molecular or biochemical pathways are critical, then flies might be the best approach. It's quicker, less expensive, but as you get more and more involved in the specifics of the importance of the brain structure and neurological symptoms that we see in the ataxias, then you have to move to I think you'll move to a mouse model, which not human, but anyway, it does, it's much closer from a brain organization and a neurological standpoint.
That's really good to know. Most of the treatments we've talked about in this talk has been something that's either injected into you, consumed. We have an attendee who's interested in more stimulation-based treatments. Are you familiar with any research using ataxia mouse models where they've looked at stimulating different parts of the brain?
No, I'm not, but I I seem to remember, and I can't unfortunately um, recall exactly where, at least in some human ataxia patients, people were exploring brain stimulation, and I think this was magnetic stimulation. Uh, um, so, given the success of stimulatory approaches in diseases like Parkinson's, it is an area under investigation, but it's outside of my area of expertise.
That is totally fair. And right now, we're trying to organize a talk with a medical doctor who looks at different kinds of stimulation technologies in humans, not in mice, but
So
hopefully
stay tuned, right?
stay
Stay tuned.
tuned. Hopefully, our schedules will align somewhere over the summer in order to bring that to y'all folks. So next, we have a question. You mentioned non-human primates in terms of that safety testing. We have someone who's asking, what does that mean? Does that mean monkeys? And
I mean,
why would
that's
you
the
use that versus
scientific
mouse?
term for monkeys. Yes.
So does mean monkey. So why would you use monkeys compared to mice for this kind of research?
I think again, they're biologically closer to humans, if you will. And the other approach about therapeutics is that you'd also like to look at the distribution of the agent in a larger in a larger brain. And so their brain size again is becomes closer to the human. And just the overall administration of the drug in a non-human a monkey, non-human primate is more similar to the sort of techniques that will be used in humans.
Along that line about techniques being used in humans, there's several drugs right now that are being tested that involve injection through the spinal cord.
Right.
We have an attendee who's wondering if all these therapies, if they will need to be injected through the spinal cord. Is this being tested in mice? Or are you also testing potential treatments that could be an oral medication or some other
Right.
kind
Yeah.
of modality?
Yeah. So yeah, this is so the way they're injected in humans, like when we put the ASO into the mouse, we went directly into the cerebral ventricle, which means we went through the brain down into the ventricle. We don't want to do that in humans if we don't have to. And the way you get it into the cerebral spinal fluid in humans is at the lower end of the spinal cord. There is a region where there's CSF but no neural tissue, these lumbar injections. And so that's where sometimes viral-based gene therapy treatments are administered, the ASOs are administered, and so forth. And all I can say is that there's an active area of research with these various different therapeutic modalities to transition them from having to be injectable to being able to be administered by oral consumption. So modifying the ASO with various chemicals such that now it can be taken up by the brain, and likewise modifying the virus so it too can enter into the central nervous system tissue. So when we think about therapeutic strategies, we're not only thinking about research to develop a therapy that targets an important aspect of the disease, but we also need to think about research on how best to administer that therapy so it's as non-invasive as possible.
That's really good to know. And also very complex, so thank you for explaining that.
Yes, yes.
We have someone who's asking, has heard about something called cerebellar organoids in order to sort of recapitulate ataxia phenotypes in a dish using cells. They're interested in your thoughts on using ataxia mouse models versus cerebellar organoids to recapitulate disease phenotype and how this could be used in this
Great
body of
question. A
research.
question that scientists yes. So these cerebellar organoids are derived from human progenitor cells that we get from ataxia patients. And so their positive aspect of them is that it's a human model system, if you will. But like the questioner said, these are organoids. They manifest some of the characteristics of the brain, but certainly not all of them. And my view is that they're very complementary to the mouse models. And I think, an important aspect of research that we're doing is some of the critical things that we see in the mouse model, we're carrying over to these human organoid models. And so if it's really a valid aspect of the disease, an important aspect of the disease, or a valid treatment, we should replicate the positive findings in both systems. So the question brings up a really important point, is that it's sometimes very important to validate what you see in the mouse models in a more human type of model system, the organoids. But again, they're parts of the brain in a dish, and so they're not. We can't put them on the rotorod or ask them to walk a fine line. So the organoids, that is.
Yeah, it sounds like getting multiple types of data from different systems and combining it together is the strongest way to go about this.
Absolutely.
And then our next question is about wearable devices and if they could be tested out in mice, or is that a human-only sort of
That's
research
really a human
happening?
only, from my understanding. When we're doing similar sorts of approaches in mice, rather than having a wearable device, there's a video camera. And so we just film the mice going through their normal behaviors. And then there are various AI programs to break down that video into normal versus abnormal behaviors and so forth. I'm not aware of any wearable devices for mice.
It might be a bit tricky with they have four legs, we only have two. Uh,
Yeah, and they beat each other. Oh, never mind.
and then we have a question. This is specifically about Friedreich's ataxia. Uh, have researchers tried directly injecting frataxin into the mutated genes in the mice? What would that
Um,
do?
I'm not aware of that approach. I'm not a Friedreich's ataxia expert, but I have not heard anything like that as a technique. The trouble with injecting protein is getting it to where it's supposed to be, and injected proteins often are readily degraded, and so forth. So
And then we currently have one last question in the chat. Do all drugs need to go through mouse model testing first and then monkeys, or non-human primate testing before being tried in humans?
all new experimental therapies do need to go through this sort of preclinical study. Uh, uh, Sometimes there's already FDA-approved drugs for one symptom or one disease that are shown to be efficacious in another disease, in which case they they don't have to go through these types of efficacy safety studies because they've already had what we call human exposure. And back to our mouse models, there are investigators that are taking already human approved drugs to see if they show any efficacy in the mouse model, because that would really, if we did get a hit in those sorts of studies, that would really shorten the time frame in getting it to patients.
Well, I don't see any more questions in the Q&A window. If you do have any other questions that are related to mouse models or other topics outside of mouse model research, please feel free to send them to us at research@ataxia.org. We sometimes get flooded with questions after sessions like this, so there might be a little bit of a delay in getting back to you, but we'll try to get to you as soon as possible. And with that, Dr. Orr, thank you so much for explaining mouse model research to us. This is a big topic, so thank you so much. We appreciate it.
What have I told you repeatedly? It's hairy.
All right. Thank you, Harry. We appreciate it.
You're very welcome. Take
And to
care.
all of you who are on this session, we hope that you learned something helpful about, well, ataxia research broadly, mouse models broadly, or both today. Wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take
Stay
care,
well,
everyone.
everybody.